This video is going to get you through the Microbial Growth chapter, which is chapter 4 in the textbook. This chapter is about how fast bacteria can reproduce themselves and what do bacteria need in order to be able to grow and thrive and reproduce themselves. And so this chapter is going to relate to lab in a fair amount of detail. First thing we do in this chapter is talk about an important person who helped discover a lot of the growth requirements for bacteria. Robert Koch is how I say his name. I'm not sure how he said it because he is of course no longer with us today. But he had been experimenting with different ways to grow bacteria in the lab, and while he could grow them, he couldn't really keep everything in its place essentially, and so he kept getting contaminated samples. And working with his wife, he discovered that he could take a compound from a seaweed that's called agar and then mix that into a liquid broth to turn the liquid broth into a semi-solid that even today we still just call agar, although agar is really an ingredient that makes the broth a solid. So we call these agar plates. The agar has a bunch of different ingredients in it, so you can have something that's just called nutrient agar, you can have blood agar, but the agar is really just what's in there to help solidify that medium. You'll learn in lab that the consistency of an agar or solid plate is a little bit thicker than Jello, and Koch is the one who helped discover that you could turn that liquid broth into this. It's a lot easier in a lot of cases to work with a solid medium than it is a liquid medium, and so that's one of the things he did, is give us that solid medium to grow organisms with. Now this picture is just to kind of remind you, agar comes from a seaweed. This is the seaweed that it comes from. Now, another part of this chapter is how do you grow bacteria in a lab? Bacteria, just like everything else, they have a certain environmental niche that they have to meet. They have temperature requirements, they have nutrient requirements, they have waste they have to get rid of. And if you want to grow them in a lab, you have to meet all the needs that they have and provide them with all the nutrients they need and remove all the waste that they have. One of the things that we do in the lab to maintain their temperature is we can grow them in an incubator. The incubators in our lab are set for 37 degrees Celsius, which happens to be human body temperature, and that's because a lot of the organisms that we study are either commensal, which means they live with you, or they're pathogens, which means they live in or on you and they harm you as they do that. And so body temperature is kind of where they grow the best, and so the incubator helps us keep them at that temperature, because your body temperature is of course not the same as room temperature. Now, describe the process of binary fission. This is very similar to mitosis, but mitosis is defined as division of the nucleus, and of course prokaryotic cells don't have a nucleus, so that means by definition they can't do mitosis. That said, they still run through kind of the same basic process. So they replicate their DNA so that instead of just having one single circular chromosome, the cell for a little bit is going to have 2 circular chromosomes. It's going to pull those towards opposite sides of the cell, and then it's going to build a new cell wall in between those 2 new chromosomes that get developed, and then it's just going to split along that wall. And so that's how binary fission works. Now when we talk about microbial growth, this is what we mean. We don't mean, generally speaking, that the cell gets bigger. We instead means that the number of cells get larger within a sample, and so it's a reproduction thing. It's not a growth and development type thing. So how does the book define microbial growth? It's an increase in the number of cells in a population. So it's just an increase in population size. Explain the concept of generation time. It essentially tells you how fast a particular species of bacteria can reproduce. Different species have different reproductive times or generation times. One of the fastest happens to be Escherichia coli, or E. coli for short. It can reproduce with optimum conditions about every 20 minutes. But then you also have some slower bacterial species. So Mycobacterium tuberculosis is again the causative agent for TB. It takes a lot longer to reproduce, somewhere between 12 to 24 hours depending on the environment that you happen to have it in. So anytime we start doing calculations about generation time or the number of cells in a population, you have to know how fast that bacteria can reproduce, and so you have to know the generation time, and that's something I'm always going to give you. What do the variables mean in the equation? Nt equals n sub 0 times 2 to the nth is the next thing that you guys have. So Nt is the number of cells in a population at a given time. Essentially that little t is the time that you have in question. N sub 0 is the original number of cells that you first inoculate into a growth medium. It's sort of fictitious. You're never really going to know that number, but I'm always going to give you that number so you can try to figure out how many cells you would have in a population after so much time. That exponent that you have going on here, that's the number of divisions that could be present, and in order for you to know that, you have to know one, how long you have been incubating your sample, and two, what is the generation time of that sample. So this is the first example that we're going to do. I think it's the example from your book. So let's say that we put 10 cells into a broth to start to grow at time 0. We are going to allow that bacteria to incubate for 4 hours, and let's say it's E. coli, that means it can reproduce every 20 minutes. Well, in order for you to figure out N, you have to figure out how many times does 20 minutes go into 4 hours, and that's 12, because you got to remember that 20 minutes, there's 3 sets of 20 minutes in any hour, and we have 4 hours, so 4 times 3 gets you 12. I have learned that a lot of students are not awesome at figuring out how many times so many minutes goes into an hour, and my advice for you is twofold. One, think about an analog watch and then try to figure out like how many times does 15 minutes go into an hour. That's 4. 20 minutes goes into an hour 3 times, and so on, because of course there's 60 minutes in an hour. If you still struggle, even after trying to picture time on an analog clock, go to the math lab. They have a person in there that can help you out with some of the math that is specifically designed to help STEM students, and that includes micro students, try to work through things. Okay, so coming back to what I had been saying, we inoculated the broth with 10 cells. Since we let it incubate for 4 hours and it had a generation time of 20 minutes, that means the exponent is going to be 12. So we have N0 is 10 cells that we put in, times 2 to the 12th power. Now, every calculator is unfortunately a little bit different in terms of how it's made. One of the things you need to keep in mind is when it comes time for a test, you are not going to be allowed to use your cell phone, so make sure you have a scientific calculator, or at least a calculator that can do exponents. When you look at most calculators, they are going to have a button that says x to the y power. And that's the one that's going to help you do this 2 to the 12th power. So at least on my calculator, the way that I do that is I hit 2, and then I hit x to the y, and then I hit 12, and then I go ahead and hit equals just to get that, and then I multiply that by 10, because that's the number of cells that we had to begin with up over here. And so that gets you this total number of cells. After 4 hours, there would be 40,960 cells present in the population. So I'm going to give you guys some additional examples of this, because you do need to be able to calculate how many cells will be present. But part of the issue here is it's really difficult to do this online. So what I'm going to say is, if you can come to a conference or talk to me about this before or after lab and you're struggling, it's a lot easier to teach this in person and show you what your calculator needs you to do. Because again, I know every calculator is a little bit different, unfortunately, but that's the basic setup for how you would calculate generation time. Okay, I gave you 2 more examples in the lab, or pardon me, in the lecture notes. First one is if a bacterium has a generation time of 30 minutes, and you start with 100 cells at time 0, how many cells will you have in 30, 60, 90, and 120 minutes? I gave you this problem because it's actually very simple. You can really honestly do this in your head, but I want you to use the equation and then use the in your head part to see whether or not you were right or wrong to get started. So I'm going to try to use this color, and I'm going to try to use this lovely new pen that I have. So this, and of course my lovely little new pen doesn't want to work. That would make my life too easy. Let's see, I've got an old pen, it's not as good, but I'll try to make it work. This right here is our N sub 0. That's the number of cells that we put in order for us to get started. So we're going to do 100 times 2, and then we need to figure out our N. So since we have a generation time of 30, if we want to know how many cells we would have in 30 minutes, that means we could divide 1 time. So we would put a little 1 right there. Well, of course, 2 to the 1 power is 2, so after 30 minutes, we would have 200 cells present in the population. Now, again, I said you could do this in your head. If after 30 minutes, that's 1 generation time, a population of bacteria can double every generation time. So we started with 100 cells, we're going to double. In 30 minutes, we're going to have 200 cells. So that's your math check to make sure that you're right. Okay, next up, we're going to do that in 60 minutes. So we still have 100 cells that started us off at the very beginning. We still have the times 2, but we need to figure out what the exponent is going to be. So in 60 minutes, this population could divide 2 times. So this time it's going to be 2 squared. Well, 2 squared is, of course, 4, and 4 times 100 tells you there will be 400 cells present in the population. I also did 90, so that's 100 times 2, this time you're cubing it, because in 90 minutes we could divide 3 times. 2 cubed is 8, so that tells us there would be 800 cells present in the population. Notice the trend. Every generation time we are doubling the number of cells in that population. So 100 cells went to 200, 200 goes to 400, 400 goes to 800. Again, you could hopefully see where we're going. The next answer should be 1600 if we're going to be doubling, but we'll still set it up down here just to make sure that we're good. In 120 minutes, we could divide 4 times. 2 to the 4th power does equal 16. 16 times 100, 1600 cells will be present in the population after 120 minutes, which is just 2 hours. Okay. After that, I gave you a problem here in the PowerPoint that is not actually in the notes, and so we're going to go ahead and do this one next, and I'm going to change my color just so you can see the different notes that I'm using for this one. Let's say you're working with Staphylococcus aureus in the lab. You weren't being very careful, and 1 cell landed on your skin because you weren't wearing gloves like we have told you to do, probably by this point at least a dozen times. It has a generation time of about 27 to 30 minutes. Now, I gave you a time range, but we're going to do the 30 minutes because the math is simpler if you do it that way. You didn't wash your hands until the end of class, 2 hours later, so how many cells would you have growing on you, assuming optimal conditions? Okay, so to set this up, here's our n sub 0. We had 1 cell that landed on us. Times 2, we need to figure out that exponent. So if it divides every 30 minutes and you didn't wash your hands for 2 hours, that's 4 divisions. So 2 to the 4th power is 16. And so that would mean you would have 16 cells even though you only started with 1 cell a couple of hours before that. Now, here's the other thing that I want you guys to think of, and it's why we talk about contamination so much in the lab. You never end up with just 1 cell landing on you if you have an accident. Like if you spill 1 drop of broth on you, you have spilled hundreds of thousands, if not millions of cells onto yourself. And if you give those 2 hours to divide, that can end up really quickly turning into billions of cells that are on you. And so because bacteria can reproduce so quickly, it is very important that you do everything that you can to avoid contamination, just because it doesn't take long for things to get out of hand. Now, I gave you another example in the notes, the Mycobacterium tuberculosis example. What I would like for you guys to do is practice doing that, and once you have your answer, you can either kind of compare what you have with other students in the class, or you can email me to find out whether or not you're right on that. I do also have a little worksheet that I usually do for my face-to-face students, and if you would like just some other examples of that to get practice on, just email me and I can send you that page, but I'm going to go ahead and keep going in the notes at this point. Alright, next thing that you have is define biofilms and provide several examples, both beneficial and harmful. So a biofilm, I'm going to look to see if there's a definition off on here, and I don't see it on that slide, so I want you to have a nice easy place to write stuff down. This right here is probably your best thing to write down for the definition of biofilm. If you're unsure of what a community is a bunch of populations in one place. So it's not just one species, it's several different species. And a polysaccharide, that's just like starch or cellulose, it's a complex sugar. And so they're wrapping themselves in sugar to protect them from their environment. Now there's a reason I chose the pictures that I did off down here. First off, there's some examples. So this is sort of a blown up picture. I don't know if you guys have ever been to Dinosaur Valley, but if you try to cross the rocks that are submerged in the water, they're very slippery. And they're slippery because they have this stuff growing on them. This stuff is a biofilm of a bunch of bacteria and a bunch of algae that are photosynthetic, all living together sort of harmoniously. They create that polysaccharide encasement to help protect them from drying off, because of course, you know, we live in Texas. Eventually there won't be very much water running through here. And so that slime coating essentially helps them stay wet for a longer period of time. And so biofilms do help create that slipperiness. That's why I was showing you that picture. Plus, just I like to look at my son back when he was adorable. He's my adorable little boy. Some other examples that I want you guys to get down. I'm not sure if I'm going to have it on a slide, but I'll come to this page. Dental plaque is one. We had mentioned that in a previous chapter. It's medically relevant because it can cause cavities in people who are susceptible. When they go get teeth cleaning and that dental plaque gets scraped off, it can actually dramatically increase the risk of developing not just sepsis, but also heart problem as the bacteria start to overgrow on heart valves, and that can lead to problems that way. So a lot of people end up having to take antibiotics prophylactically before they go to the dentist to prevent that heart valve problem that can happen for them. Some ear infections have been proven to be biofilm, so again, not just one species, but lots of species of bacteria growing in the ear. People with cystic fibrosis, if you don't remember what that is from previous classes, it's a genetic condition. It's autosomal recessive. What that usually means is two parents who didn't know that they were carriers, they have a kid who ends up with cystic fibrosis. The people with cystic fibrosis make this exceptionally thick mucus. It's way thicker than anything you guys are familiar with, and it's very hard to clear it out of the system, especially the respiratory system. If you don't remember what mucus is, that's just a protein, and a protein is just a nutrient. And so all these bacterial species come and start living on that mucus in people with cystic fibrosis, and they end up with crazy frequent infections of both the respiratory system and the digestive system because of the nutrient that they were providing all of those bacteria. So it can cause complications for people with cystic fibrosis. We use biofilms in water treatment. That's a process that I might have a picture of that. I feel like I do. So there are all these little settling tanks and stuff, but the one that says it has a biological filter, they spray bacteria on usually like some little ceramic pellets, and then the bacteria help degrade the waste that goes through there. So again, not all bacteria are bad. We wouldn't have clean water if it weren't for bacteria. And then some bacteria can also be used to help remove toxic waste like oil. If you remember when the Deepwater Horizon thing happened, oh my God, that seems like forever ago now at this point, but when that happened, they would go out and spray what just looked like water, but they were spraying bacteria that can actually eat the oil off the surface of the water. And so that's an example of using a biofilm for bioremediation or cleaning up things for you, removing toxins. All right, we pretty much said that. So next up, what is a pure culture? A pure culture means you are growing one species within whatever container you are using to grow them. So it means that you didn't contaminate your culture, essentially. Why do we use them in a lab? Because if we're doing something in a lab, we're trying to learn something about usually one species. And if our sample is contaminated, we don't know, did the E. coli use this nutrient or get killed by this drug, or was it the contaminant that actually accidentally got into our sample? So we always try to grow things one at a time. So one petri dish will have one thing, a different petri dish will have another thing. It just helps us make sure we're not confusing any of our experiments as we go through them. From a medical standpoint, we want to obtain a pure culture to figure out which one species might be causing a problem in a person who has an infection. That concept is becoming a little bit more archaic at this point, and that's because we're learning that a lot of infections aren't really caused by one species, they're caused by a lot of species, those biofilms that we were talking about just a second ago. Next thing that you guys have is explain aseptic technique, and I say that you'll get more of that in lab. By now we should have talked about that in lab. But these are all the procedures that you do to minimize the chance that you contaminate any of your samples. This involves wearing your PPE so you don't accidentally breathe on your sample, because by now you should have learned breathing on a sample means you have just exposed additional bacteria, viruses, and possibly fungi into the sample that you're working with. It's cleaning your glassware beforehand. So we use the autoclaves to sterilize our glassware before we put anything into it. Or sometimes we'll put the broth into it and then sterilize it, and ends up doing the same thing. But sterilizing things so that we know we only have one species growing in anything, that's another aseptic technique that we have. Flaming our loop between transfers, that's another aseptic technique. So it's everything that we're doing to try to make sure we are maintaining a pure culture in the lab. Next up, you have define a bunch of terms. Colony. So there's a bunch of colonies you can see on this plate. Here's a colony. Here's a colony. A colony is a mass of usually millions of bacterial cells in one place, and a colony is visible on an agar plate or on a slant, even when you have a slant going. Colonies do only form on solid media, so you will not see colonies on broths or in broths, so you have to have that solid medium in order to see it. Now, we used to always be told one cell started a colony, but now sometimes we know that multiple cells can land in the center, and then we call that a colony forming unit. But I still, because I'm old and it's what I was taught, I usually talk about the fact that a colony was descended from one cell, because that's how the majority of them seem to start, although there are complications to that sometimes. We already talked a little bit about agar before, but agar is a polysaccharide that is derived from algae that helps to solidify the media. Again, it's what makes our broth into a solid, is what I said earlier. Petri dish is what we put the agar into in some cases. So Petri dish has a bottom and a top. The agar gets put into the bottom. Hopefully by now we have learned whenever you incubate these, you incubate them upside down so that the bottom is on the top. That helps to prevent that condensation on the lid from dripping down and drowning the bacteria that we have on the plate. One of the questions that I ask you here is the Petri dish airtight? No, it is not, and it's important that it's not. As it turns out, bacteria breathe just like you do, and they need oxygen as well. And so while we do want to prevent contamination, we don't want to prevent air from getting in there so that the bacteria can actually breathe. When we say agar plate, we just mean a Petri dish that has agar in it. And then next step, you have explain or define the streak plate method. Streak plate method is something that hopefully by now we have done in lab, and it's something that you're going to do for your unknown later on. So when a patient comes in potentially having some type of infection, you collect a culture from them, or really you collect a sample that you're going to grow in a culture from them. But a person doesn't just have one thing growing on them, they have a bunch of different species. A streak plate is a way for you to isolate one species out of the mixture that you actually got from your patient. What you do to create a streak plate is you dip your inoculating loop after you have flamed it into the sample that came from the patient, and then you zigzag across the plate a few times. Then you flame your loop to sterilize it again. The next few steps, they're all designed to reduce the number of cells growing so that eventually you're going to see isolated colonies. So this was the first plate. This person streaked their plate, and notice you can't see individual colonies anywhere in here. It's just a lawn of bacteria. So step 2 of the streak plate, after you have flamed your loop, is you're going to drag your inoculating loop across your original streaks a couple of times and then zigzag again in another quadrant. That reduces the number of cells that you pull. So notice how this growth is not as dense as we had in quadrant 1. This is quadrant 2. After quadrant 2, you flame your loop again, then you drag across here and you zigzag again. Sometimes people go ahead and go for a 4th one, and in fact when we teach you in lab we want you to do a 4th one just to give you a better chance to do it. By the time you're done streaking your plate, again, you should have isolated colonies like what we have right here. Remember that colony is one species that are derived from one original cell. We can then start to test those to figure out which one of these might be causing the infection in the person, and then figuring out exactly what species that was causing the infection in that person. So that's the streak plate and what it does. It helps you, from a mixture, isolate one species from that mixture. In our lab, we grow bacteria as batch cultures. What does that mean? It means we have to constantly transfer our organisms from one thing to another, and usually in our lab we grow things in slants. This is what a slant looks like. So after a couple of weeks we'll transfer from an old slant to a new one. We have to keep upping our batches or renewing them so that we always have new cells in order for us to pull from. Some labs don't have to do things in a batch culture. They can just keep new bacteria growing in this big vat by adding more media and removing old dead cells, but we can't do that. We don't have the very expensive machine that helps them do that. Let's see. Describe the stages of growth that occur in a batch culture like what we have going on. So when we first inoculate any sample, now when we start talking about this, we're going to be talking about a broth. Similar things happen on a solid medium though, but when you first inoculate that broth, the bacteria at first don't really start to divide. They survive, they don't die, but they don't start the process of binary fission, and that's because usually we're taking them out of the refrigerator to inoculate a new medium, and so they're cold. They also usually have a bunch of waste built up, and that stuns them just a little bit. And so that first phase right after you transfer them, it's called the lag phase. They're not reproducing, but they're not dying either. They're just kind of sitting there like, holy crap, what just happened? Because we moved them to a new home. After the lag phase, what happens is the bacteria realize we just put them into a new place with a bunch of nutrients, and so they start the process of division, and you get a huge explosion in the number of cells in a fairly quick time period, depending on how fast the generation time is. When I was taught this, it was called the log phase. Your book at this point is switching more to, they call it the exponential phase, but it's the same thing. Log or exponential growth is the exact same thing. What's important about the log phase is a couple-fold. One, that is when generation time gets measured. So when I said earlier that E. coli can reproduce every 20 minutes, it can do that in the log phase. It doesn't do that in the lag phase or the stationary phase. It only reproduces every 20 minutes during the log phase of its growth. It's also the time when bacteria are most susceptible to antibiotics because they got so excited that you put them into a new place that they aren't protecting themselves anymore. And so this is when you have the best chance of murdering them all off with antibiotics if this was an infectious sort of a situation. Next up, stationary phase. What happens here is they're starting to run out of nutrients in the medium that you're growing them in. So some of the cells are starting to starve to death, but other cells are doing okay and they're still reproducing. So what's happening during the stationary phase is the birth rate and the death rate are equal to each other, so the population stagnates and the number stays the same, but we do have birth and death. It's just that those things are equal. After the stationary phase, you get the death phase, so the death rate increases past the birth rate. And then you get this really weird phase. What happens is it's practically impossible to kill all of the bacteria in a sample because they will start to cannibalize their neighbors. And so you get this phase of prolonged decline that I'm going to come back to here in just a second. Let's see. Next, you have which stage is generation time measured. I said that earlier. It's the log phase or exponential phase, whichever one you guys want to call that. I'm perfectly fine with either one. Which stage is most important medically? It's the log phase because, again, that's when they're most sensitive to antibiotics, so they're easier to treat during that one. There's a couple of other little things that end up becoming important during these phases, and it has to do with the chemicals that the cells are actually making. Primary metabolites are small molecules that a cell can make as they're in the process of multiplying. So it's things like amino acids. If they're fermenting, it's stuff like alcohol. Their basic nutrients are things that are just being made as the cell is living their life, but they do tend to be small molecules. Primary metabolites are made during the lag phase and the log phase until you start to get towards the end of the log phase. Then they switch over towards something else. And those something else is what you have next to define on your thing, in your notes. So secondary metabolites are different. These are chemicals that cells start to use to outcompete their neighbors. Secondary metabolites are like antibiotics. As it turns out, a lot of antibiotics are actually made by other bacterial species so that they can murder their competitors and have an advantage in terms of survival. So that's what a secondary metabolite is. They accumulate later on, so towards the end of the log phase, the stationary phase, and especially that prolonged decline, that's when we start to make those secondary metabolites. During which stage does the statement survival of the fittest hold the most meaning? I have this particular picture I want to use for that. So this is more of a realistic phase of prolonged decline. What happens, and we haven't really talked about strains just yet, but there's more than one kind of E. coli. There's different strains of E. coli. Some of them can produce different toxins. Some of them can use different nutrients. They're all still E. coli, but they're different, if you kind of want to look at it that way, races, but they're called strains in the bacterial world. Well, what happens during the phase of prolonged decline is, during this phase, this one particular, hey, look at that, it's the exact same color. Anyway, this strain does really well at first, but then another strain comes up and it has started to make a toxin that can kill that first strain, and so this one starts to die off, and then this one starts to win. But then another one comes in and it starts murdering this one off, and so another one comes in. And so they're really just kind of murdering their neighbors and trying to figure out who can survive the best and the longest in this environment where it would be great if somebody would transfer us to a new medium, but we've been stuck in this stupid tube forever and nobody is transferring us. So survival of the fittest is in response to strains. Some of the fitter cells start to multiply and some of the weaker cells start to die off, essentially. All right. Next, you have how does growth on a solid medium differ from a liquid medium? So in a liquid medium, because of diffusion and osmosis, everything ends up being mixed together fairly well. So there's the same number of nutrients at the top and at the bottom. Everybody's in the same boat. Here in a solid medium, though, when you're talking about a colony, you've had this cell in the middle here for a long time, and so it's been eating the nutrients that are in that agar for days. So the cells that are in the middle of the colony start to die off, but these cells that are around the edge, well, they just got to new agar where there's still plenty of nutrients and there's very little waste, and so these cells start to do great. And so basically, depending on where a cell is in the colony, it may be struggling to survive or it may be thriving because now we don't have an even mixture of nutrients. There's a partitioning of resources that happens because we have a solid medium. All right. Next up, we have different temperature requirements for different species of bacteria. And temperature is what we are going to be starting out with. There's other factors that are going to play a role here, but To start us off, anytime you see that word extremophile, it tends to mean that something can live in an environment that we didn't know something could live in. So something that's really hot, really cold, it's a really harsh environment. Usually if something is an extremophile, that's going to mean it's an archaean. We had talked about that a little bit earlier, but that's just a word that we use. Extremophile means they like extreme conditions. If it helps you out, remember phile means you like and extreme means extreme, so they like extreme conditions. Okay, next step, psychrotroph. Here's what you need to know for a psychrotroph. Now, 20 degrees to 30 degrees Celsius means basically room temperature is where they grow really well, but they can handle refrigeration temperatures too. They don't grow as fast when you refrigerate them, but they do still grow. And so what I wanted you to know about them is they are important for food spoilage. This is why it's important to not let your leftovers sit in the fridge for a month and then try to eat them. It's because these guys can still grow at that temperature, just more slowly. Next one that you guys have that you need to write some stuff down is mesophile. These guys like it a little bit warmer. Notice that their temperature range, humans are right around here in their temperature range because human body temperature is about 37 degrees Celsius. And so these are the ones that are pathogens. Every pathogen is a mesophile and they like your temperature. Thermophiles like it even hotter than that, so 45 to 70 degrees Celsius. Remember that 100 degrees Celsius, that's boiling, so they're not quite that hot yet, but they still like it really hot. You can find these in hot springs that aren't boiling. These aren't pathogens. These are just things that are living in like compost piles or in those hot springs that I mentioned. There's other groups, but these are the only three that we're going to be talking about right there, so just make sure you have that. How does refrigeration help preserve food? It slows bacterial growth. Again, it doesn't prevent it, but it slows it down so that your food stays edible for longer periods of time. What is average human body temperature? 37 degrees Celsius. It is important that you know that you have different temperatures in different places. Hopefully in AMP you learned about core versus shell temperature. Your core temperature is warmer than your shell temperature is. Like it's a different temperature if you measure it rectally versus you measure it orally or a forehead measurement. Same thing kind of works here. Degrees Celsius has a range, but usually normal body temperature is somewhere between 96 and 100 depending on where you're measuring it and what's going on with that person. Provide examples of organisms that cause disease in the extremities because they need a cooler temperature than your human body temperature. The two that I'm giving you pictures for here and examples of are leprosy, which is what the woman on the left has in this picture, and then syphilis, which is what the person, I can't tell the gender from that picture, has. That's a canker. From syphilis. Used to be one of the ways that we would treat syphilis is we would on purpose give a person malaria because malaria makes a person spike a fever and syphilis can't handle that fever. Syphilis used to be one of the leading causes of death. So if you were going to die anyway, what's a little malaria on top of that? So that's why we would use that treatment option prior to the invention of antibiotics. I do want to go ahead and give you the names of these guys. Now, Treponema pallidum, that was a model organism in an earlier chapter, so go back to that to remember how to spell it. Leprosy is Mycobacterium, just like Mycobacterium tuberculosis. This is a Mycobacterium too, but it's leprae, L-E-P-R-A-E. That's how you spell the leprae. Next, we're going to talk about oxygen requirements for bacteria. If you see that something is anaerobic, it means that it needs very little or no oxygen. Some anaerobes, even a tiny bit of oxygen will actually kill them, and so you actually have to handle them in very specific environments. Obligate versus facultative. I want to kind of look at those words for just a second. If you see that something is an obligate, I want you to think of it as they are obligated to do that. They must. So if you have an obligate aerobe, it must be aerobic. It must have oxygen or it will die. Hold on just a second. Let me turn my ceiling fan on. Okay. You are an obligate aerobe. If you don't have oxygen, you will die. There are some bacteria that are like that as well. Micrococcus luteus is a species that we use in lab. It's an obligate aerobe. It must have oxygen. Facultative. It means it can, but it doesn't really want to. So a facultative anaerobe would prefer to be aerobic and have oxygen, but if there's not oxygen, it can switch to fermentation or anaerobic pathways. So they can survive that way, but they would prefer not to. A good example of one that does that we've played with in lab is E. coli. Yeast, Saccharomyces cerevisiae, is also a facultative anaerobe, so hopefully you know yeast can be used to make alcohol like beer and wine. It does that only if it switches to a fermentation pathway. if yeast is allowed to use aerobic pathways, it won't generate that alcohol. And so it can do either or. It depends on what you want it to do and what's available for it. Obligate anaerobes, those are obligated to stay away from oxygen. Those are the ones where even a little bit of oxygen will actually kill them. A good example of that is Clostridium botulinum. That was a model organism from earlier. That's the one that causes botulism, that paralysis that we talked about earlier. Microaerophiles, these are like the Goldilocks. They want a little bit of air, but not too much air. If you give them too much oxygen, they die. Give them too little oxygen, they die. So just a little bit. Helicobacter pylori is a species that lives in your stomach and can cause ulcers. It's a microaerophile. Aerotolerant is like the opposite of Goldilocks. They do not care. They can have oxygen, they cannot have oxygen. It just doesn't matter whether or not there is oxygen available to them. Underneath that, you have explain how a shake tube is used. And so this is where that picture at the bottom is going to come in. A shake tube can help you figure out which of those oxygen requirements a species actually has. So what you would do is you would heat up some agar and a broth and have it be liquid, and then let it cool off just a little bit, enough that you can inoculate it with bacteria, but not so cool that it starts to solidify. Once you inoculate it and it's still a liquid medium, you shake it up. After you shake it up, then you let it cool, and then you wait to see where does the bacteria grow in the solid medium after it has cooled off. If the bacteria only grow at the very top, that means they're obligate aerobes. They must have oxygen. They can't live down here where there is no oxygen. Facultative anaerobes, this picture doesn't show it as well as I would like to, but what it would look like is there would be more dots up here at the top. You would get more growth towards the top, but you would still see some growth down at the bottom. They prefer it up here, but they can survive down here as well. Obligate aerobes, you would only find those at the bottom where there's no oxygen. Microaerophile, that's the Goldilocks, so you find them somewhere in the middle where there's a little bit of air oxygen but not too much. And aerotolerant, same concentration everywhere because they don't care where there's oxygen and where there's not. It would be a good idea to kind of doodle that picture and this explain how a shake tube is used. My note says that picture is on page 101 in the textbook, but I don't know what edition I took that note with, so I don't know what page it's on anymore. It might still be on the same one. All right. Next up, what is catalase? And we'll eventually test for this in lab, know which organisms are catalase positive and catalase negative as mentioned in the text. What is it? It is an enzyme that can break down hydrogen peroxide. This is the chemical formula for hydrogen peroxide. It's H2O2. When you mix it with catalase, it splits into water, H2O, and then molecular oxygen, which is O2. Now, you are a catalase positive organism. Hopefully you know that if you drop hydrogen peroxide onto a wound, it bubbles. That's because you make catalase and it splits the hydrogen peroxide, and the bubbles are the oxygen that comes off of that. Now, those species of bacteria that are aerobic and use oxygen, they have to deal with some of that oxygen, and so they will very often produce catalase so that they can deal with this that gets produced in the process of all of their metabolic reactions. So we can actually test to see whether or not an organism uses that or not. So what we would do, and this is just one way to test it, and I don't even think you're not going to do it on an agar plate like this. You're going to do it on a slide instead. But you would take some of the organism, you would smear it on a slide, and then all you'd have to do is drop hydrogen peroxide on. If it bubbles, like this one is clearly bubbling, you would know that the organism is catalase positive. An example of a catalase positive one that the book mentions is Staphylococcus species like Staph aureus and Staph epidermidis. If it does not bubble, then it is catalase negative. Streptococcus, like Strep pyogenes that causes strep throat, it is a catalase negative species. Those are the ones that your book gives you. Okay, next we're going to switch gears and talk about something else. So far we've hit temperature and we've hit oxygen requirements. What we're going to talk about now is pH requirements. The pH of our agar has to be maintained within whatever range that species is that we're trying to grow. So a neutrophile likes it somewhere around neutral. Remember that a pH of 7 is neutral, but they can handle a little bit outside of that range. So there's where a neutrophile tends to grow. Acidophiles like it more acidic. Remember that anything with a pH less than 7 is technically acidic, but I'm going to say, and I'll even take the less than that, anything less than 5 is going to be an acidophile, and anything greater than 8 is going to be an alkalophile. They're going to like a basic environment. Next up, we're going to talk about water availability. A couple of terms that are going to be important for us, and I have mentioned these terms in a previous chapter. Plasmolysis means that the cytoplasm has shriveled away from the cell wall. So this picture right here is showing you plasmolysis, versus this would be a turgid cell. If you plasmolyze a bacterial cell for long enough, it will kill that cell. One of the things that you can do to initiate plasmolysis in any cell, not just bacteria, is you can expose it to a hypertonic environment like super salty water. If you put, let's say, a bacterial cell in the super salty world that is anchovy, it will plasmolyze the cell and kill it. That's why there's so much salt in preserved food. It's so it will plasmolyze bacteria and be fresher longer on a shelf, if you want to call it fresh. Halotolerant is a species that can tolerate up to 10% salt. So that would be a species that, I don't actually know what the salt concentration is of an anchovy, but let's say it's 9%. It would mean it could actually grow inside that anchovy container. A species that is halotolerant is Staph epidermidis. That's a species that lives on your skin, and your skin can get to be pretty salty, especially if you've been sweating a lot. Halophiles, they like even higher levels of salt. We used to think the Dead Sea didn't have any organisms growing on it because it was too salty, and as it turns out, there's a bunch of halophiles living in there. Usually halophiles are in that domain archaea that we had talked about a little bit earlier. Let's see. Next up, I thought I had a model organism for you, but I don't really see it on here at this point. So I guess I'll just go ahead and do it. Model organism is Thermus aquaticus. It used to be called Thermophilus aquaticus. What type of habitat does it derive from? It comes from hot springs. That's actually what the Thermophilus meant originally, was they were a thermophile, they liked it hot. Does it cause disease? No, you're not as hot as a hot spring, they can't grow in you. Using some of the terms from this chapter, discuss this organism. It's a thermophile, it likes it really hot. How does this organism relate to DNA fingerprinting? We haven't talked about DNA fingerprinting yet. We will for later materials, but that's how, like if you've ever watched those CSI shows or whatever, they'll tell you whether or not the DNA matched at a crime scene. There's a specific reaction that you have to do prior to fingerprinting. It's called the polymerase chain reaction, or PCR. Well, the polymerase chain reaction, it's an enzyme that we actually got from Thermus aquaticus that helps us replicate DNA during PCR, the polymerase chain reaction. So how does the organism relate? The organism gave us the enzyme needed for PCR, which happens before DNA fingerprinting. All right. Next up, we're going to talk about the elements that bacteria need. Now, some bacteria actually need organic molecules just like you do, like they need carbs and proteins and lipids, but some of them, they're good with just elements on a periodic table. And even the organic molecules, of course, they're just made with elements. And so we're going to take this all the way back to just what elements do these guys need. The first 6 that you need are right here. These are the major elements. I'm circling more than 6. Hold on. 1, 2, 3, 4, 5, 6. There we go. Those are the first 6 that you need to know. Here's what's special about those first 6. Those 6 elements are what are used to make the organic molecules. Like carbohydrates and lipids are made out of carbon, oxygen, and hydrogen. Nitrogen, you can find that in both nucleic acids and proteins. Sulfur can be found in proteins. Phosphorus can be found in some phospholipids, which make all cell membranes, including bacterial cell membranes. It can be found in ATP, which is the energy carrier that not just your cells use, bacteria use ATP as well. And you need phosphorus to make nucleotides as well. So those get used to make all 4 categories of organic molecules that you should have learned in your freshman bio or your A&P class. The next 4 that you have that I've kind of scribbled through at this point, potassium, magnesium, calcium, and iron, these are usually found as ions within the cell and within the body. They are usually used to help enzymes out, and so they're usually cofactors that help with enzyme production. Now, another thing that I want you to do, and so I'm going to switch colors if it will let me, which apparently it doesn't want to let me because my computer just hates me. I want you to put a star or highlight phosphorus and then iron. Those 2 elements are considered to be limiting nutrients. What that means is those are usually the elements that are present in the lowest amounts in nature, and as a result it slows down the growth of bacteria because they run out of those nutrients and they can't continue to grow after that. And so that's one way to help control bacterial growth, is limit those nutrients. Let's see, anything else I want you to know through there? Not really. All right. After that, we've got some other terms that I think I kind of start with right here. Anytime you see troph, T-R-O-P-H, it has to do with eating or how a cell gets everything that it needs in order to survive. Heterotrophs, they eat organic carbon, like they have to have carbohydrates or lipids or something along those lines. Autotrophs, on the other hand, can eat inorganic carbon, because auto means self. They can make their own organic molecules. So they can just use carbon dioxide and then make organic molecules out of that. You, incidentally, are a heterotroph. Plants are autotrophs. Okay. Carbon fixation is the next word that you guys have to define. That is the process of taking inorganic carbon, usually in the form of carbon dioxide, and then converting it into an organic molecule, usually some type of carbohydrate, although it can switch away from that. Saying that a different way, carbon fixation converts inorganic carbon to organic carbon. Nitrogen fixation. Now remember, nitrogen was needed for us to make any of our proteins and our nucleic acids as well. Well, the most abundant form of nitrogen that's in the air around you is molecular nitrogen, that's N2, but neither plants nor you can use that kind of nitrogen. So what has to happen is, and only bacteria can do this, they can convert molecular nitrogen into a form that is more usable, either ammonia or nitrate or nitrite, and that conversion is called nitrogen fixation. Saying that a different way, nitrogen fixation is conversion of N2 to ammonia, and only prokaryotes can do that. What are limiting nutrients? Phosphorus and iron. That was actually the second part of that question. Which elements tend to be limiting? Phosphorus and iron. Back to the definition of what they are. They're available at the lowest concentration relative to need. So again, there's not very much of that available for organisms to use. You have, what are the trace elements next? And I guess I blew past that. Here are your trace elements right through here: cobalt, zinc, copper, molybdenum, and manganese. They are required only in super teeny tiny amounts, and I'm talking about micrograms of those things are what's needed for bacteria to grow, so teeny tiny little amounts. What is a growth factor? It is something that any organism needs in order to reproduce itself. You guys have growth factors. If growth factors aren't present, your cells won't go through the cell cycle. Bacteria have those as well. It's just something that they need in order for growth. Do all organisms have the same growth factors? No, it's different for everything. Even between different people, there are different growth factors that are necessary for those. Another important thing about a growth factor is you can't make those for yourself. You have to obtain them from the environment, usually in a pre-made form. Next up, was fastidious on this one? There we go, it was. Fastidious. If you have a fastidious bacteria, it essentially means you have a very picky one, where you have to provide it with every single nutrient because it can't make anything for itself. The way your book says it is, it has complicated nutritional requirements. The example for that is Neisseria gonorrhea, which is the causative agent for gonorrhea. You have to grow it on blood agar because it needs so many different nutrients, and really only blood can provide it with all of those different nutrients. All right, coming back to different types of trophs. Photo means light, so a phototroph gets its energy from light, usually sunlight. Chemotroph, it gets its energy from chemicals. Now, we can take the photo and chemo and put it together with the auto and hetero that we talked about just a little bit earlier. And I want to say I have a table of that. Of course I don't have a table of that. That would have made my life too easy. So I'm going to create the table. And I'm going to try to make this neat so that you guys can do it in your notes. Okay, over here on this side, we're going to be talking about how does an organism get its energy. You've got two options. You can get your energy from light, or you can get your energy from chemicals. Now there's prefixes that get used in relation to that. If something uses light, the prefix for that is photo. So in both of these boxes beside light, we're going to write photo. And hopefully you can write neater than me on a computer screen. For the chemicals, they get the prefix chemo. Then, I don't even know why you came up. Across the top, we're going to talk about how do they get their carbon. Do you get it from inorganic sources like carbon dioxide, or do you get it from an organic source? If you use inorganic carbon, you get the next prefix auto. And so that's going to go into all the boxes below that. If you use organic, that was supposed to be a G, organic ones get that prefix hetero. I'm sorry, I'm really messy. It's really hard to write on a computer screen, y'all. And then the end of every single one of those is troph. I'm just going to abbreviate it with a T. So here's what that means. And what did I start with in the notes? I started with photoautotroph. So photoautotrophs get their energy from light, and they can use inorganic carbon as their carbon source. So again, plants are photoautotrophs. There are also some species of bacteria that are photoautotrophs. The one that I want you guys to write down as an example is a group of bacteria called cyanobacteria. I probably have that written on the previous slide. There we go. There's how you spell cyanobacteria right there. So cyanobacteria are like the bacterial equivalent of plants. They do photosynthesis exactly like plants. They even have the same chlorophyll that plants are going to have. There's even a hypothesis that the chloroplasts present in plants is really just a cyanobacterial cell that kind of got trapped inside the plant cell and it became a chloroplast over time. Okay, next up you have photoheterotroph. I've got the definition for that off over here, but we'll come back to this. So photoheterotroph uses light as its energy source, but they have to have organic carbon in order to survive. These guys are weird. There's only a couple of examples of plants that do, or plants, pardon me, of bacteria that do that. And one example is a group of bacteria called purple non-sulfur bacteria, which unless you've been to a swamp, you have never run into. But if you've ever seen purple snot-looking stuff on the surface of a swamp, that's purple non-sulfur bacteria, and it's an example of a photoheterotroph. Okay. Next up, I'm just going to call them chemoautotrophs, but your book adds a litho, which just means rock, because they're getting some of their nutrients from rocks, specifically phosphorus. For chemoautotroph, though, that's this little group right here. They're getting their energy from chemicals, but they can use inorganic carbon for their carbon source off down there. And again, these are really weird guys. They live in places that you can't go to. I think I have them on the previous slide. This is a hydrothermal vent community. It's a place where volcanic superheated water comes out at the bottom of the ocean. We only discovered that they even existed back in the 1970s. They are coated with these mats of bacteria that can actually essentially eat the sulfur that's coming out of that superheated water. And so, really weird. Didn't even know they existed until, oh my god, that was 50 years ago at this point. I am so old, y'all. Okay. Last example, chemoheterotroph, this one. They get their energy from the chemicals that they eat, and they must have organic carbon. You are a chemoheterotroph, so are all the pathogens that can cause disease in you. The example, oh darn, that I'm giving you down here at the bottom, this is Pseudomonas aeruginosa. It is a species that we're going to play with in the lab just a little bit. It can cause opportunistic infections, especially in burn victims, which is what has happened up over here. But it's an example of a chemoheterotroph, just like every other pathogen happens to be. All right. Next, these are the tables that can help you kind of separate out what we were just talking about and the nutrients and what they get used for. So this is just all the stuff we just talked about in more detail. Next up, what is a complex medium is what you guys have next to the notes. A simply complex, it means you've piled a bunch of ingredients into one thing to try to grow bacteria. The way your book says it contains a variety of ingredients, but it's relatively easy to make. The examples that we are going to talk about, that we are going to use in the lab, include nutrient agar and broth, chocolate agar, which does not have chocolate in it. It's actually just broken blood cells. McConkey agar, Thayer-Martin agar, and blood agar. Those are all complex medium. Next, chemically defined. This is a good study table for all the things that we're about to talk about. It gives you examples. We are going to use blood agar, we are going to use McConkey and nutrient agar. We don't use the other ones actually in our lab though. I already said complex media, so we were good there. Chemically defined is sort of the fastidious broth. You have to measure out every single nutrient that you're going to put in there in very precise amounts. You use these for specific research that's happening in the lab. The example that you guys are getting for that is glucose salts, which again, we don't use any of the chemically defined media in the lab, but you have to measure out every single one of these ingredients separately and then mix those all together to actually make your media when you're doing a chemically defined medium. Next, what is a selective medium? It's a medium that essentially selects for some species to grow, but kills off other species. So it's a way for you to isolate a particular species. Why would it be used? Because it makes it easier to isolate a species. Some examples, we are going to be using McConkey in lab. Well, this is kind of like McConkey, but it's not. We are going to use EMB, which is eosine methylene blue. There's another EMB off down there. We're not going to use HEC, but we will use EMB and McConkey agar, which your book mentions. Thiamartin is another example of a selective medium, but we don't use that one in our lab. What is a differential medium? It makes different organisms look different as you see them growing on the medium. So I mentioned McConkey earlier. McConkey is a complex medium, it's a selective medium, and it's a differential medium. It allows some organisms to grow bright pink colonies, and others just look like regular colonies grown on it. So it makes different organisms look different because of the ingredients that are actually in there. Blood agar is a complex medium, and it's a differential medium as well, because some bacterial species are able to break the red blood cells down, and you get something called hemolysis. And so you can see them grow. I shouldn't use red as I'm drawing on blood agar, apparently. I have to switch colors again. But this is beta hemolysis. Only some species can do that, and so it makes them look different if they are capable of breaking down those red blood cells. So it's an example of a differential medium. Underneath that, we have to define the different kinds of hemolysis that can occur. So beta hemolysis, that's the clear growth. And what I mean by clear is it actually kills all of the red blood cells and destroys them so that the agar itself looks clear. And you can see through it on that side. So you get a clear zone of hemolysis is what I want you guys to write down for beta hemolysis. Alpha hemolysis is what you see right here. And this picture doesn't show it quite as well as I want to, but what happens is you kind of mostly break down the red blood cells, but not completely. So instead of clearing them out, you end up with essentially blood cell poop, and it kind of looks greeny brown as the growth. And so alpha hemolysis, you get partial clearing that ends up looking sort of greeny brown instead. All right, so we talked about different oxygen requirements that different species had, and I mentioned that there are obligate anaerobes that oxygen can kill if you try to grow them. So one of the ways that you can grow them in the lab is to create an anaerobic environment, and this is the cheaper version. And even the cheap version, this stupid box costs $600. It's ridiculous. But what you do is you put your plates in here, and then you activate this gas pack, and the gas pack sucks all the oxygen out of the environment, so you can grow your anaerobes inside that container. Old school way to do that was with a candle jar. You literally lit a candle inside the jar, and the candle consumed all the oxygen so that it created the anaerobic environment. So that's the cheap way to do this, because who wants to spend $600 for a plastic box? The expensive way to deal with anaerobes, and you might have seen people using these in some different TV shows, when you see them sticking their hands into what looks like a glorified fume hood, that's an anaerobe chamber. There's no oxygen inside of there, and you have to stick your arms through these little gloves, and so you're working with the organisms inside that environment while you are out here where there's oxygen still nice and happy. All right, next up we're going to talk about different ways that you can count how many cells might be present in a sample to see how contaminated something might be or how much growth is actually present on a particular medium. This table is kind of what we're trying to recreate in the little table that I gave you in the notes. For direct microscopic count, here's what you need to know. It is the fastest way for you to get cells, but you do need about 10 million cells per milliliter to actually view the cells under the microscope. A limitation of a direct microscopic count is you do count both living and dead cells. You can't tell what's alive and what's dead, so you end up counting them both. Cell counting instruments, you just run a broth through this little essentially capillary tube, and this device measures the shadow, and so it counts the cells that go through here. Limitation, exact same thing. It's counting both living and dead cells. It can't tell the difference between them. A plate count uses a process called a serial dilution. So what you do is you have a broth that has billions of cells in it. You put a milliliter of that into 9 milliliters of usually something like normal saline, something that's isotonic to the cells, and then you keep diluting it, and then you plate out a milliliter of each one of these, so you're going to have 4 plates at the end. And the reason you do that is your plate has to have between 30 to 300 colonies to count it, and you're not sure which one of these tubes is going to give you that count on a plate. Let's say that this one did right here. You do the math, that's a 1 in 100 dilution, so that would tell you if you counted 30 cells, you have to multiply that by 100 to figure out how many were in the original cell culture that you got off over there. So you count the colonies that are actually growing on the petri dish after you've done your serial dilutions. The limitations for this one is it only counts living cells, and you have to incubate before you can do your count, and incubation takes anywhere from 24 to 48 hours, so it's a slower process. Next up, you have turbidity. Turbidities can be run whenever you're growing something in a broth. The more turbid, like notice this is very turbid off down here, the more cells there are in that area, versus the less turbid an area is, the fewer cells you have in that area. This is really just a way to estimate, like this one has the most growth versus this one has the less, unless you're also correlating it with a plate count. And so your limitation for this is, if you want it to be accurate, you have to correlate it with a plate count. Acid and gas production. We're going to be doing phenol red tests in the lab, and this is a way for you to test it. First and foremost, I do want you to notice this one is more turbid than this one. Notice you can't see the blue lines that went behind that, and that's because it's turbid on this side. But going back to acid and gas, this was a phenol red broth. It was red before we put the organism in, but because the organism fermented the sugar that was in there, it changed to yellow, and that's because it produced an acid. So we had that pH indicator that is phenol red that told us we've definitely got growth over here because we changed colors. The limitation for this is it detects growth, but it really doesn't quantify. So you don't know how much of an organism is in there. It just tells you there is an organism in there. All right, so that completes that chapter. Make sure you complete the assignment in Schoology for